Hello everyone, welcome back to the class. In our previous class, we had started discussing about plastids. So plastids are double membrane bound and they are semi-autonomous organelle. So they have been originated from endosymbiosis and they are present in all plant cells and photosynthetic protists. So these plastids are having multiple copies of double-stranded DNA as genetic material and 70S ribosome for protein synthesis. So the plastids will always differentiate from proplastid. The plastid will be differentiated, and the plastids which differentiate from proplastid, that collective term is used as plastid. So proplastids are inherited with cytoplasm of plant cells. So this proplastids acts as a progenitor cell. Proplastids develop according to the needs of specialized cells into chromoplast, leucoplast and chloroplast. Chromoplast is having different types of accessory pigments other than green color, so they help in pigment storage. Leucoplast are colorless plastids and they store starch or fat. Chloroplast are responsible for photosynthesis and they possess chlorophyll pigment. So proplastid differentiate into chloroplast by some internal changes. First, proplastid may or may not have inner membrane. So this proplastid forms the inner membrane, and that inner membrane will fold, and that will further fold into discrete disc-like structure to form thylakoids and grana, and also it forms stroma thylakoid, forming the chloroplast. The chloroplast is double membrane bound structure which is responsible for photosynthesis. This proplastid will differentiate into chromoplast, where it is having accessory pigments necessary for color in fruits and flowers. Proplastid will also differentiate into leucoplast, which is a colorless storage organelle which stores starch. That the plastid which stores starch is amyloplast, and there is another plastid which stores fat, that is known as eleoplast, and there is another plastid which stores protein known as proteinoplast or alluroplast, which stores protein. So these were the plastids which differentiate from the proplastid, which we discussed in the previous class. So today we will be discussing about transport of proteins into chloroplast. So plastid is a double membrane bound semi-autonomous organelle present in all living plant cells and photosynthetic protists. So they have double-stranded DNA and 70S ribosome, and plastids differentiate from proplastid. So proplastid, the collective term used for different kinds of organelles derived from proplastid is plastid, and it is inherited with cytoplasm of plant egg cell. So they become chloroplast, leucoplast and chromoplast according to their need. So if we have to define the structure of chloroplast is, so the chloroplast is having outer membrane, inner membrane, intermembrane space, and there are structure known as stroma, which is the fluid filled region, and there is a stack of stacked sacs which is known as granum, and each flattened sac is called as thylakoid. That thylakoid is having photosynthetic pigment. So grana are interconnected by unstacked stromal lamellae. So there are lipids found in thylakoid membrane. About 80% of lipids are uncharged mono and digalactosyl diacylglycerol, and only 10% are phospholipids. So this was the endosymbiotic theory of origin of chloroplast. So etioplast, where proplastid develops into etioplast in dark grown plants which have the precursor pigment protochlorophyll instead of chlorophyll. When plant is shifted to the light, or when there is exposure of light, so etioplast rapidly change into chloroplast by converting this precursor chlorophyll. So transport of proteins into chloroplast. So these chloroplast proteins are synthesized on cytoplasmic ribosome and are imported into one of three chloroplast membrane. Here, as similar to the mitochondria, there is three location: outer, intermembrane space, and inner membrane. So the targeting of proteins takes place in three destinations. So there is a particular sequence to which protein is adhered. There is N-terminal signal sequence. That sequence is known as transit sequence. Which target chloroplast protein to the import machinery in the outer envelope. And there are two types of complexes. That is TOC and TIC. TOC means translocase or translocone at outer envelope, and TIC means translocone at inner envelope membrane of the chloroplast. So the targeting begins by binding of transit sequence, that is protein which is having transit sequence. That sequence binds to the outer membrane receptor, that is TOC 159 and 34. These TOC 159 and 34 are GTpases. They belong to these receptors of members of family related to guanosine triphosphate. So they need GTP hydrolysis process for binding to the transit sequence. So they form, so after binding transit sequence with the TOC 159 and 34, they form ternary complex, which stimulates the hydrolysis of GTP. GTP hydrolysis happens and transfers the transit sequence to the next translocone. So After forming ternary complex, so this protein, precursor protein with the transit sequence is transferred to the next translocon, that is tick, by forming a continuous spore. If this is stalk complex and this is tick complex, it forms a continuous spore across both membranes for translocation into the stroma. So let us see the flow chart. Transit sequence target chloroplast pre-proteins to two outer membrane receptors, stalk 159 and stalk 34. Both receptors are members of the family of related guanosine triphosphatases with overlapping function. Bound GTP favors binding of transit sequence with TOC 159 and TOC 34. It forms a ternary complex of transit sequence with both TOC 159 and TOC 35, which stimulates GTP hydrolysis and transfer the transit sequence to the translocon. GTP hydrolysis may open the translocation pore or promote binding of TOC to tick to form a continuous spore across both membranes for translocation into stroma. Next, the beta barrel protein TOC 75 is a prime candidate for the channel across the outer membrane. So it is a prime candidate. The pore seems narrower than those in the alpha helical seq translocon. So polypeptides are thought to be unfolded during transit. Then, so now where the protein is, so protein which is having transit sequence has crossed TOC and it is in tick. Now what happens? The pore across the inner membrane consists of a complex of at least 7 tick proteins. The abundant protein tick 110 not only forms some or all the pore but also binds the protein to HSP70, which is a chaperone on the stromal side of the membrane. So what does this HSP70 do? HSP70 by hydrolysis of ATP in both the intermembranous space and stroma promotes translocation of imported protein. After it is translocated to stroma at last. So it is translocated to stroma. Now protein is in stroma where there is the last procedure happening, that is cleaving of the transit sequence by signal peptidase. And before the signal peptidase cleaves, the proteins are folded or redistributed to their final location after the cleaving off of transit peptide. Then some proteins fold with the help of HSP70, HSP100 or HSP60 based on their destinations. So let us see the flow chart now. The pore seems narrower than those in the alpha helix seq translocon. So polypeptides are thought to be unfolded during transit. The pore across the inner membrane consists of a complex at least 7 tick proteins. The abundant protein tick 110 not only forms some or all of the pore but also binds HSP70 chaperones on the stromal side. So ATP hydrolysis by HSP70 in both intermembrane space and stroma promotes translocation of imported proteins to the stroma. As protein emerge into the stroma, signal peptidase cleaves off transit peptide before the protein folds or redistribute to their final location. Some proteins fold with the help of HSP70 and HSP100 chaperone and an HSP60 chaperone and remain in the stroma. Other proteins move on to thylakoid membranes or the thylakoid lumen using at least 4 different pathways. Some photosynthesis proteins insert directly to the thylakoid membrane. So after coming inside the stroma, the protein has 3 fates: thylakoid, thylakoid membrane or thylakoid lumen. So some move across the thylakoid membrane into the thylakoid lumen through the translocon. So hydrophilic proteins destined to the thylakoid lumen will retain secondary N-terminal signal sequence after the transit sequence is cleaved in the stroma. As the protein enters into the stroma, there is cleavage of the transit sequence, but the proteins which are to be translocated to the thylakoid lumen will retain the secondary N-terminal signal sequence after the transit sequence is cleaved in the stroma. So after translocation, the peptidase in the thylakoid lumen removes both type of secondary signal sequence. See, hydrophilic proteins destined for the thylakoid lumen retain a secondary N-terminal signal sequence after the transit sequence is cleaved in the stroma. Some move across the thylakoid membrane into the thylakoid lumen through a translocon. Other proteins with tightly bound redox factor cross the thylakoid membrane while compactly folded using translocon factors. After translocation, peptidase in the thylakoid lumen removes both type of secondary signal sequence. So this is the image which shows the translocation of protein into the chloroplast. So here the transit sequence binds to the 2 outer membrane receptor, that is TOC159 and TOC34. TOC34 is associated with GTP hydrolysis activity. Bound GTP favors binding of the transit sequence. So there is formation of ternary complex here, that is transit sequence TOC159 TOC34 stimulates GTP hydrolysis and transfer the transit sequence to the translocon. GTP hydrolysis, so here it is closed. GTP hydrolysis opens up this translocation pore and helps to promote binding of TOC with the TOC with the tick to form continuous pore across both membranes of translocation into the stroma. And here you can see HSP70. HSP70 is a chaperone which is present in the stromal side of the membrane. ATP hydrolysis by HSP70 in both intermembrane space and stroma promote translocation of imported protein still inside the stroma. After entering into the tick complex, so the signal peptidase removes the transit peptide before the protein is folded or is distributed to their final location. Some proteins like HSP70, HSP60 remain in the stroma to ensure the protein has completely folded. So in our next class, we will be learning about peroxisomes. Thank you.